this course will have some integral component in practice. You need to have some clarity what we research mean, because probably from next semester, even for you people, I think mini project is already there, right? You have already done some projects in your BTech program, but you have possibly you have followed some rules, guides, instruction, and you come up to some conclusion, finding out some intuitive results. That is all fine. Now there is some clarification, some classification of research is also there. We will also look into that. What are the different kinds of research we can do, and which domain our research, particularly biological people, biotechnology or allied field, what kind of research you are going to have. That classification is also important. So I'll show you some of the important aspects along with the textbook, what you should follow, because I have shared you two textbook definitely, but all contents are not in a same manner, right? I have shared, I'll just show you what is the component we are expecting from which textbook. So this is okay. So I have already shared you this textbook, right? You can see research methodology from Kothari. Okay, CR Kothari. This book is fine, but very point wise. Like it looks like you have to study, you have to memorize. Like that kind of approach is there in this book. Not really intuitive, but organized, because they have many sections, subsections. So, for example, in today's class, you may please note that we are going to nearly discuss about the chapter 1, which is talking about the introduction to the research methodology, because this is of course a part of this curriculum. We will definitely discuss them, but not necessarily in the same line as they are talking about motivation of research, meaning of research. There is no really clear-cut boundary when I'm speaking about objective and motivations, right? Objective is for short-term goal, you already know. Motivation is bigger one. We normally say beyond your monetary values. What can be the logic behind that? If I ask you objective of choosing your MSc program and motivation behind MSc programs are definitely different. You may have some inclination to this subject, you are more interested maybe there. These are all motivating you to come over here. And so once you are here, what should be your objectives? The schedule, the plan, maybe I'll say a specified terms you are going to attain through this process. So this way, the definition, you see, you'll always find some definition 3 4.5 wise, like that. You need not follow very strictly on this book. So first chapter I will suggest we'll discuss, actually that's why I'm introducing you. What is the meaning of research, what is the objective, what different kinds of research are there, because when I'm talking about research, you are talking about everything, including the biological science, medical science, including social science. This is also research, right? Even if we are not doing, it is a part of this process. Significance of research, method versus methodology, research process, criteria for good research. You will always understand, once you are a good research, you will understand what should be the desired criteria. Of course, this comes from some our intuition, some components we will discuss from the textbook. So please keep in mind. So for example, this Kothari textbook chapter 1 we are going to discuss for today. Next day probably will be going a little bit more on how to explore the design, how to make a design. All those concepts will come in chapter 2 and 3. In the same manner, here we'll also discuss, not now, because the first step is here philosophy of natural science. Okay, that is the list, the Elsevier book, right? I already shared with you, research methodology in biological science. Peter Lake. So this book 1.1 unit will be having some discussion today, not much details. And then in coming days, we'll have research methodology, strategy and planning. This is very important here. And this section I will definitely cover in our next class. Now, without much introduction, let's start what we are going to discuss today. Of course, we have some PPT. PPT is just to make me orient and you also uh, follow the track. Not necessarily you should mug up this PPT and questions will come. Not that sense. Okay. So coming into the slide, so from this point, when I'm going to ask you what is research and how you, one second, someone has joined. Okay. Uh, so if I ask you first definition of research, of course there is no unified definition. What is research? What is not research? That things comes from your understanding. Now, what is your inception about research? What do you think a research can be, and what cannot be research? Those questions are obviously, if I ask 10 people, even my 10 colleagues, the answer will be definitely different. Okay, but unequivocally we can say, when we are talking about research, what is research is definitely looking into perspective of a theme or a domain or a problem. Of course we are having interest, we know this is the problem, we need to identify, this is the part of the research, not definitely. But when I'm doing in a manner, so that what is the conclusions or what is the outcome of this sorry, what is the outcome of the study can be proven in a definitive manner. So for example, if you are going to identify one phytochemical which can help to reduce your blood sugar, uh, is it research? For example, I am injecting some phytochemical and you are finding that blood sugar level in the patient is coming down. Is it a research? No, this is not a research. Then what could be the research? If I'm going to say that some compound I'm injecting or I'm taking as a dosage form and proving something, so I'm trying to correlate. Okay, so I'm trying to correlate between two things. One is what is the way we are going to plan the study. So not necessarily I'm having some 10 people, one people, for example, I'm giving the doses form and I find blood sugar level is low. Is it sufficient to say? No. I need to know what was his uh, blood sugar level before administration, after administration. Is there any change? Yes. One people is sufficient? No. I need to go for a large number of population, so that my conclusion is proven by some evidence. So evidential proof is really required whenever I'm talking about doing a research. Now, you are a very fast timer, you are going for a research and you are collecting some literature from textbook and maybe internet. You are downloading many articles on use of this plant in, for example, sugar management in diabetic people. In that case, is it research? No, because you are simply accumulating some information from the information retrieval systems or from literature. This is not a research. This is going to be a review. Review is kind of things you already know, right? Review is kind of things we are making out something from the past knowledge we already have. So for example, last 20 years whatever things has been done, I am just accumulating, I'm making a compendium so we can see it later on. This is not a research. So research should have definitely one protocol, one objective. It should have a logical sequence of experiments, and the results we are getting should prove your assumption. Of course, when you are doing some research means you have something, some intuition is there, either to prove it or disprove it anyway. So either you are saying yes it can reduce, or you have to say no it cannot reduce. So this way we can define a research topic, right? So please highlight this terminology when I'm talking about systematic approach. That is really important. Systematic means if you are doing a research, your friend is doing a research, and I am doing a research, all should follow a systematic approach. How should we go to tackle this problem? How I'm going to analyze this? How I'm going to add something and see the relationship, that is called casuations, right? Connecting two things. I'm putting something and I'm getting some observations. That means we are connecting addition of this thing is going to give result of blood sugar level decrease. So that is called graduations. So this is a simple observation. Maybe your drug is not working at all, simply the person is having some other disease and that's why his blood sugar level is falling down. So you need to have concrete design which will prove your hypothesis beyond any doubt. That part is really important. Obtaining and confirming new knowledge. This is really a trouble for newcomers. If I ask you to do some research, many times you do not really see whatever has already been done. Of course, you are not the first person to do something, maybe. Okay, we are assuming in academic research, you are not maybe the first person to do something. Some people, maybe 10 people, 20 people, 100 people, 1,000 people have done significant amount of work on this domain. Let me tell one example. One of my PhD scholar is doing a research on polyhydroxyalkanoate or bioplastic production using microorganism. Is it anything new or novel? New or novel, I mean to say, novelty will come from when it is going to be reported primarily for the first time, not been reported some observation, some phenomena, some mechanism, whatever we are saying, it has not been reported earlier. So that gives a novelty edge. It is also a good point for the researcher. But thing is that when the domain is so much explored, maybe last 20 and 30 years, some 1,000 people, more than 1,000, 2,000 people have already worked so many different kinds of work on polyhydroxyalkanoate production. And I'm giving my student do some study. What she is doing, she is collecting some microorganism, she is inoculating with media, because you are biological people, that's why I'm speaking this example. And she is isolating the polyhydroxyalkanoate from the microorganism. Is it a research? No, people have already done. People have already done. So what can be your intuition? Okay, if she is doing with some new microorganism, nobody has done this, and she can find a much higher productivity. Of course, it is a research. She is looking for new species. She is looking for increasing the yield. So somehow his or her work should stand alone, which should confirm, and it should give some new knowledge, reliable knowledge. Reliable is very important aspect when I'm talking about research methodology. This part is also really important. Reliable means by chance you are getting something is not a proven science, right? Because based on your science, next generation people or maybe the other people will also concentrate whether this hypothesis is correct or not. They will try to implement on that. So your results should be reliable. Reliability comes from repeated and standard formalization of your methodology. So if I'm using a methodology which is very significant, which is very unique, which is going to measure only pHa, there is no other called confusion or maybe biasness, then I'll say the result is reliable. So C should have a reliable, not only reliable, it should be reproducible results. Then we will say this is going to give some A's on research. Systematic, orderly, right? So that thing is there, and purpose is to get some new knowledge, something new. Now you may say, sir, I got a new microorganism. Is it not new? Of course new. So this is of course a research, but there is some more problem. People look even more novelty in your work, only getting a new microorganism. This is a research, I agree, but may not be the whole population. Scientific community may not be simply interested to find a new microorganism, right? You got my point? Because today you are finding something new microorganism, tomorrow another people will do and find a new microorganism. So new microorganism, microbial diversity, you know, right? It is so big. So finding some 10 or 20, and if you continue year wise, you will definitely get some new microorganism which is producing. So simply you are not going to do anything, you are not having any plan, simply you are taking some 1,000 soil sample and try to isolate something, and after that you speak nothing why you are getting more, For example, if you say that I'm getting more microbial species from wastewater, then you connect why we are getting more microorganism in wastewater. If you connect those things, that is a systematic and a new knowledge you are going to provide to the society. Of course, this can be a research part. So this is a general approach we are talking about, not necessarily biology. It can be any other domain. It can be social science. It can be in business or management people. They can also have systematic, orderly manner they will do, and they will get something new out of that. This is all about research definition. So essentially, you should have a careful investigation. Uh, when I'm giving you these points, please try to remember. You need not memorize those sentences. You need not memorize word by word. Nothing like that. Only understanding point of view. You are giving some points so that to look more legible. You can follow. Research is thus an original contribution. Originality is really high important word. Okay, other words are there to make a sentence. Okay, here is a careful investigation through search of new facts. You can do the search of new facts through literature review, as well as doing some experiments you can do, and literature you must check. While how to check the literature, all those aspects we'll be discussing in coming days. So that will give you some observation, you can have some comparison, and doing some experiments you can attain, you can pursue the new knowledge in this domain. Okay, objective and systematic methods to finding a problem. And what is not a research? Can you say what is not a research? Suddenly you discovered something, but you are not proposing any mechanism. Okay, that means you are lucky enough to get something. That is, we cannot term it as a research until or unless it is stemmed from some hypothesis. When you are going to do any research, some hypothesis should always work. Okay, for example, I am going to explore wastewater for some new microorganism. Why we are going to look for new microorganism in wastewater? I have intuition that wastewater contains so many of different diversity of compound, right? So microorganism growing there could have a capability, inborn capability, or maybe they have mutated, or they have made a substantial change in population, so that which microorganism can survive there, which can take up those pollutants, can grow there. Okay, so many times you will find many people are looking for isolating a microorganism, for example, from contaminated soil. Okay, for example, chromium, not chromium, I will say some pesticide degrading microorganism. People will try to isolate from pesticide contaminated soil. It's a logic, right? Because you already have intuition, as the microorganism is thriving in this community, they might have the microorganism which is having the ability to degrade pesticide. Probability of getting there is more. So this intuition is there, and that's why you are looking for a new microorganism. So systematic getting some information is not sufficient. You should have a logic. But so you try to understand accidental discovery. Okay, accidental discovery may occur in structured research process. So if you're having some plan, you can have, otherwise it is not going to make a new things. Usually it takes a form of phenomena not previously noticed. It may happen that this thing you are not shown, but suddenly you are going to get some new phenomenal changes in this process. But when it may lead to some structured process to verify and understand the observations. So it may lead, if I'm having those points in together and all comes under a accidental manner. Okay, accident is not really a research. You are going to look for microorganism which can produce, which can degrade pesticide, and suddenly you discover no, it is not degrading pesticide. For example, it is degrading halogenated hydrocarbon. Okay, so that kind of presentation, that kind of material, when I'm proposing, when I'm placing, it cannot come into a research criteria. Data collection, simply I'm going to get some data. Of course, data collection is the important part, but simple data collection may not be. So data collection means you are compiling, for example, last 20 years, how many people have reported what microorganism is producing PHA. That data, is it a research? No, at least not a research. It can be part of some review. You are combining all those things. So that's why when you say about scientific publication, you already know, right? There are different kinds of publication. Do you know that? For example, you are doing some research, you are not responding at all. You are doing some research. So what is the final objective? If you are doing some research means you will try to find out something new, and as a researcher your final objective will be to publish somewhere. Publish means, what is the reason people publish, do you know?
So there is a proof
To
that
get
they've done the
recognition.
research.
Yes, the first part is
Yes,
getting
like
recognition. As a researcher you are spending some hours in lab and finally nobody recognize you. It may not be really good for you, right? Everyone knows, everyone wants to get recognized in their own domain, right? So when you are spending time, you want to have recognition. To want to have recognition means your work should be publicized, people should know what kind of work you are doing in a broad audience, right? If you are not doing so great work, then what you will do, you will try to present in your department level some conference. If you are doing moderately good, then you will go to some national level conference, where your work somebody will at least appreciate. You are doing extensively good work, you can go for international presentations. You are doing so, depending on how much nobility, how much depth of work is there, you can go in different level. You can do for publications. Publication can be in, that we will discuss later on, not now. You can have journal publications, you can have book chapter publications, right? Conference publications. You can have some tabloids or maybe some magazine, newspaper. Everywhere people wants some extent of recognition. So that is always the driving force. So you need to always understand what is going to contribute. You need to always publish your work, and to make a publicize your work, your work should stand apart from all other, right? So you are going to collect some literature, and this is called a review. Of course, it is a publication, but this doesn't come from research. So your all publication can be classified, whether it is a research article, whether it is a review article. And nowadays many publisher has different subcategory. Okay, full length research articles, that means a complete set of work. Okay, you can have something short version of your research, very small but very important work you are publishing, very short work. You are doing a big project, part of the project you are publishing, it is happening, right? So this kind comes from, for example, if I'm going to have this kind of publication, so short publication, right? Short research articles, or sometimes letters. Many journal publishes letters, very short, 3-4 page scientific understanding, scientific discussion on the topic. So anyway, so these are all part of important aspect, collecting data to get reliable knowledge. These are all part of research, but itself cannot be a research. So searching out published research result in library or internet, you see, we are not classifying it as a research. But it does not mean that it is not recovered. It is recovered. I'm giving you a topic means you have to search the internet to get those articles. So this is always an important step for research. So always it includes in synthesis and analysis. You will try to analyze what is already been done, what people have done, where they have explored. So this exploration process need. But reviewing simply literature is not a research. Okay, this is work. Okay, you should be appreciated. You will get recognized for your work, but we will not claim it as a research here. So research, what is it? Is a explanations for event, phenomena, relationship, causes, how this thing is happening, why it is occurring. Okay, how, why, what, all those questions they will show there. If there is any interaction between one phase to other phase. So planning of research is really important. It is a process planned, managed to take informed decision. It is a process of something creative. It's circular, right? Circular means whenever I'm doing some study, in any research article, if you see, they will always put some questions also. Okay, we have done this work, but this cannot be, because you have to criticize your own work. So when I am criticizing own work, what is the fault we found? Those points are also leading to new domain of research. These are the new area where you can explore more. Okay, fine. Now, when a failure to see applications can be due, so if I'm not getting any significant way to find out application of my work, what is the purpose? Why you are not getting what you are expecting for. So here you see user not trained or experienced enough in the specialized methods for your research, not only economic, because this was for economic point of view. Researchers often do not provide adequate interpretation and guidance application of the research. That is very important failure, I will say. I'll not important, a major failure. So you are doing some study and you are not highlighting what is the importance, why you should do this, why it is important to general community. So researchers are responsible to help users understand what is the implication, how. Okay. So for example, you are looking for now, for example, researcher working on discovery of some COVID-19 vaccines, right? So simply if some, I should not name any organization. For example, some organization is proclaiming they have identified some medicine which is going to work like a miracle on people, right? So do you think, do you believe that? No, because you have to give the implications. Implications means what will happen, what is going to happen. Implication of this study, when I'm putting some people, putting on some group of peoples on those vaccination protocol or then the trial, what is going to expect from that? What your expectation? Okay, are you going to stop the disease? Are you going to cure the disease? You have to clarify, you have to elaborate how much people is going to get cured from the disease with your intervention. If I'm not taking this, what is going to happen to those people? So in many cases, um, these study are actually involved, and it may give you, if I'm going to do a real research, you need to give the implications also, what is going to happen, how it is going to happen, those questions should be addressed. So when I'm talking about objective, so what is the objective of the research? If you say, you try to understand what is the objective, to know something better. And you are not knowing, people throughout the world is also knowing, because you are going to publicizing your work, you are going to publicize your view. So try to highlight these things, achieve some new insight. New insight means new mechanism. For example, you are finding some new mechanism. Okay, for example, you are exploring, you are trying to find out whether this hand sanitization is important in COVID transmission. If I ask you to verify, what you will do, you try to understand one thing. There are different kind of research design, I'll talk later. Now I'm asking you simple questions just to know how you are understanding. I'm giving you a problem, right? Because people from the very beginning are telling whether using of alcohol based sanitizer is always good, using face mask is always good. And you are a researcher and you are trying to understand whether it is good or bad, what you will do, and what is the problem in this kind of
Research
research.
on the effects of alcohol.
So what you will do, those people, so maybe for example, if you are doing the research in a lab, then what you will do, you have some 10 fellow friends, right? You will distribute the 10 into two groups, 5 5. Do you suggest the 5 you use alcohol, and you will suggest the other 5 you do not use alcohol? Then you will ask them to roam around the city, and after 1 month or maybe 2 months you will count how many people got infected. Can you do that? No.
No, sir.
No. So all research is not really experimental, right? So some research you cannot have any control. So what you will do then, you will try to find out the data. For example, I am going to find out how many sanitizer bottles are sold in state of West Bengal. I'll collect the data, how many people got infected, how many population got having this COVID-19 other state. So maybe I'm collecting the data for some 20 or 25 or 30 states, maybe different country also. I'll collect what is the increase in use of sanitization, and what is the proportion of people getting corona infection, maybe for example transmission rate. That means in this kind of study you cannot really have your own way, right? You do not have any control of the data. So you are synthesizing some information to have some new insight, of course in not in a controlled manner. These kinds of research are not in your hand. These are not really lab oriented research. This is all collecting data, and from the data you are trying to get information. So this is a part of research design we will discuss in coming slides maybe. You have to portray accurately the characteristics of the particular individual, situation, and group. Okay, what does it mean? Here you have to characterize, right? So you are doing a study with laboratory animals. That is still allowed, right? With ethical clearance, we had from animal ethical clearance we have in our institute. You can do some study with rat, mice, guinea pig. You are testing some drug effectiveness. So what you are doing, you are inducing some disease with them, right? They are not inborn disease they have, right? You are inducing disease first, then you are injecting something, some drug you are going to test. What you need to do, if I say that out of 10 mice I injected this much of drug and all 5 recovered, okay, is it sufficient information you are giving to the people? No, you have to give complete information, particular individual. So you have to say we took mice, for example 20 mice or 30 mice, each of the mice is about body weight of some 50 gram or 100 gram. You should give average body weight, their age, how much old is the mice, at what condition you are treating them, what you are feeding them. All information is required, then only the same study can be done. So what is the genotype of this mice you are using, what feeding regime you are using? That means you need to give a complete characteristics of the individual. Individual does not mean the human, right? Any subject, any subject. For example, I'm doing some soil analysis, then I have to characterize about soil. What is the basic property of the soil, which soil you are dealing with, what you are going to extract, at what condition you are going to extract, is there any other groups are involved, you are treating some soil with acid, some soil with base for extracting some metal, environmental example I'm talking. Everything should be given. So objects in view are all descriptive research studies. You should have those components. Determine the frequency which something occurs with which is associated with something else. This is very important. It is known as causes and cause effect kind of things, right? Like for example, you are exposed to some cold environment and you are getting some viral infections, for example. So these are all the hypotheses, right? Maybe some people have already proven. So you need to give some causation theory. Is it connected? And what you have to do, you have to plan the experiment such a way that yes, whoever is connected with these points, only one variable I'm changing. For example, I'm changing only temperature and they're getting this infection. Not necessarily they're getting infection, that's why the temperature is falling down. So cause and effect you need to identify, and that's coming from your research. You can really correlate the observed phenomena with the change. Okay, I'm taking this food and it is increasing body weight. I'm taking this food, it is increasing the metabolic rate. So there is a cause, there is a effect. Of course, one individual is not sufficient. You should have large number of people so that your confirmation is more strong. You can really tell. Hypothesis of a causal relationship between variable. I told you, na? One is cause and another is effect. So you can test the hypothesis. Yes, the drug can reduce blood sugar. How? You have to do the experiments. Cause is I'm injecting the drug. Result is I'm seeing reduction in blood sugar. So one is input, another is output. You have to correlate. Yes, it is happening, or it is simply nothing is happening. In one case, blood sugar is increasing. In one case, blood sugar decreasing. 10 people blood sugar does not change. Is it giving any concrete evidence? No. So to prove that or to disprove that, you should have to conduct the research. So this can be one of the important objectives when I'm talking about this one. Now, classification, right? I told you there can be different class of research. Classification means some research can be only analytical research, some is all theoretical research, some is about basic science, understanding the phenomena, some can be applied research. So this way research is also classified. So we must define the different kind of research, different criteria used to classify each type of research here. So for example, you are talking about descriptive versus analytical, right? Descriptive research is for getting the information which is already available, which you are seeing it through the phenomena, having little control on that. Analytical is going to measure something, you are going to do some changes, you can find out the values, it can be turned in a numerical perspective. We all tell them as analytical. Applied fundamental, you know, right? How, for example, you are discovering a vaccine, right? And you are going to know that what gene is involved or what surface protein this vaccine is going to neutralize. You want to know the mechanism. Okay, so if you want to know the mechanism, if you want to know the pathway, right, knowing pathway is important to the patient? No, patient it is not important, but for science it is important because it will lead to discovery of something else. So some people only do on the fundamental aspects. For example, you have found out one chemical which can neutralize the surface protein, okay, for COVID-19. If you do this study, of course, it is fundamental research till now, because until or unless it is going to generate, it is going to implement on some trial or maybe animal studies where you are looking for application orientation, it cannot be treated as applied one. Applied is for applications. Application not only for human, it can be for environment, it can be for society, it can be for animals. Some use somehow the quality of some individuals planet or ecosystem is going to improve. If you are going this kind of directions, you are going to put some study on that. This is always a applied. Quantitative and qualitative is really good or bad, this kind of sense. Sometimes when you represent qualitatives, that means we are not going to measure in a numerical pattern. Many observations can be non-numerical kind. Many are quantitative. So basically, when we are talking about biological science, most of the data, most of the analysis are quantitative. So we are not really in other side. It is a quantitative research. Sometimes it can be a qualitative when the individual can vary, right? So perception, someone is saying this is good, someone is saying this is bad. So is there any scale? Can I compare his good and another person's perception of good? No, these are not comparable. So here each good scaling is not similar. They are not same weightage. But quantitative, 2 gram per liter of, for example, your microbial culture is producing some 2 gram per liter of ethanol fermentation. 2 gram per liter is always quantitative. You are doing here, you are doing in US or Japan, your quantity of the outcome or the results remain relevant, right? It does not change with settings. Conceptual and empirical. Conceptual and empirical is you need to understand. For example, I'm going to find out any phytochemical which can be going to treat some wound healing, right? So when I'm talking about empirical means, what is empirical? Okay, some new compound will be there and we will try to find out whether something is happening, and then we will try to go into it. That is empirical. Conceptual, if you know that some already existing knowledge is there. Okay, for thousands of years people are using this compound for this plant for wound healing. That means there is a background information with you. It is a concept, and you already know this extract contains these phytochemicals, and you already know that phytochemical can actually work as antioxidant, and producing it can actually reduce the inflammation. So your hypothesis, your work is connected with some dots. So that's why you are using this one to do the study, right? They're all connected in a chain. If you know, of course, that chain is not very strong. It can always fail. Some all phytochemicals may not work. You might not have phytochemicals in all extract. Phytochemical means, for example, classification of the phytochemical. All things may not be there. So depending on this persuasion, your work can be either conceptual one or empirical one. Empirical means just observations. You do not know how is it happening. You do not have any control. You do not have any understanding, at least till now. That is called empirical.
So, but just now you said that if we just see the data and everything, then it's not a research. How can you then say empirical is research?
No, you see, empirical means you are doing study. Empirical means, for example, you are doing study. You are doing in a lab,
So we
but
are studying already the content which is people have searched, right?
Mhm. No, for
So
example,
that's not research.
conceptual. Okay, I understand what you say. People, whatever already has done, and you are simply summarizing it in a report. That is not a
Hmm.
research. That is a review. Fine. Now when I'm talking about conceptual means, you are also getting this what people have already done, what, and you are connecting the things. Okay, these plant species was utilized, these plant species contain this, you are getting from literature. And then you are connecting, okay, then this can be used for the study, and you are not simply hypothesizing, you are doing the study in lab, and you are trying to verify whether it is really working or not. Then I will classify it as a conceptual one, because you are doing, of course, for your lab work, even conceptual study, applied research, whatever research, you should have always literature review. All research will have a literature review, but literature review is not the research part. For example, now if I'm asking you to do some study, you are at working at from home, right? If I ask you to do some study on this, what you will do? You will only do literature review. You cannot really do, you cannot really perform at least wet lab
Yes.
experiments. If the lab is for biotechnology people, you cannot do that. So you are not doing a research, you are going only accumulating the data on which you can build some hypothesis, and you have to verify. You have to verify. If I am verifying this in lab and you have some strong hypothesis, I will term it as a conceptual. I will term it as a empirical if you have no strong reason to do the study. There is no background, you do not know that this plant was used. Sometimes many people will find in plant science phytochemicals, some new plant they are going to explore for this disease. It cannot be a conceptual research until or unless you can really correlate why you are using this. If you cannot tell, but you are getting good result, okay, there are two aspects. You chosen a new plant, you are finding some good result, but do you think it is a conceptual one? No, it is empirical. You suddenly got it, you are reporting your study. Oh, okay, it is a research, but there is no strong background why it should work. Then it is empirical one. Now you are publishing your work, then you are going into lab further and you are analyzing what was the reason why this thing happened. You are now connecting, and then you are moving back from empirical to conceptual research when you can correlate the reason behind that. Are you clear? You got the point? Okay. So
Yes.
basic applied, anyway, these are
Yes,
all
sir.
discussing about the things, you can go through it. Now you see, we are talking about this diagram is not necessarily you have to understand, but you have to see the connection here. So we have some basic research. Basic research is only about knowledge. We are going to connect what is the mechanism involved. It can be in different science, and we are already having some scientific knowledge, we can take it from here, we can also give some knowledge. So basic research means you are developing something, you are going into mechanistic view, you want to see something is happening, why it is happening. If you are going into looking this mechanism, how you are looking this mechanism, of course you are already trained, you already have the subject matter knowledge with you. If you don't have the subject matter knowledge, you cannot go, okay, you cannot go into this basic research. You need to know the mechanism why it is happening. So this science is already known to you, so this is going to interact with the current research domain, whatever area you are working. You are synthesizing some new information. Yes, you are telling this product is going to make these changes in the surface protein or something else, whatever may be your topic of interest, and you are giving some scientific information, new information you are proving. So that is coming into your subject matter knowledge for the new researcher. Now these are all working in a domain of here basic research. When I'm talking about applied one, so this knowledge is going through involvement of technology, human resources, institution. They're going to know and they're going to interfere with this problem solving. So that means I'm this knowledge whatever we are having in institutional framework or research framework using technology whatever is already existing, you are going to analyze the problem. So here we are adapting these things for your research work, you are going to modify, you are going to change, and this information knowledge is going to transfer the new information to other people. It can be utilized in an advancement of the research which is already going on in applied science. This is the use. So this is scientific knowledge we are gaining from basic science. Some new knowledge is synthesized. This knowledge is coming to applied research domain. So applied research, basic research, these classifications are actually based on what is the value, what is the intuition, immediate application, right? You got my point? So if him having a immediate application of some knowledge, we'll term it as um, applied research. I think I will problem solving research. These are a classification only. Analytical and descriptive, you see, descriptive research, the attempt to determine, describe, identify something. Okay, analytical will give you some more information in some measurable manner. Okay, we should know, attempt to establish why something occurs and how it came to be there. So mechanistic view is already there. Methodology we'll discuss next class. I think we are already little late. Anyway, I'll share these slides with you, and you follow the textbook, uh, whatever I'm suggesting in chapter wise. Try to understand the basic things. No need to memorize all the concepts. No need to memorize. You will understand definitely. Sometime the distinction is not so clear, right? When I'm talking about differences between textbook, what are the different kind of research is there? If I ask you, research can be different kind, right? You can have from questionnaires. Many people you will see who are not working in biological domain. Maybe they are doing some social research. People will only go for interview, right? Mostly interview, or maybe they are posting their questionnaires in social media, getting input from you. That is a research, because you are synthesizing some new information, what perception is happening. This is also research, but in a social science view. So their research methodology is different, their method is different. They will make the questionnaire. Question is very important for them. For you, if I'm assuming that you are working in a biological field and you are going for lab oriented research, your study design will be something different, your research work will be different, your question will be different. So that differences what kind of research is done is always a different perspective. Now I'm doing a study on COVID-19. I want to know how the transmission is happening, right? So what is my case? Do I have any control? No. What has already happened, that data I'm taking and I'm trying to give some view. Okay, so this is always about a posterior research. We cannot have all the data at the home, or we cannot have control on it, how the data is coming up. Only what you are doing, from the data you are trying to see, you are trying to model how the disease is happening, or what is the disease mechanism, pathogenicity, epidemiological study. If I'm going to do that, research is always different. So classification of research, if I see in the textbook orientation, there are many sub classification. Some classification will actually fall with one another, not clearly distinguishable things can be there. You got my point? If I'm taking this pandemic data and I'm making a model about how the transmission is happening, okay, or I'm projecting what will happen in 2020-21, how many people will die in India, like that. From the data I'm making a model and doing. What kind of research is this one? Is it a lab oriented work? No. Is it applied? Yes, because if I'm projecting something and I'm telling that yes, in 2020-21 so many people will die or something, and if the government is considering the research so seriously, they will take some initiative, right? So there is a real application in this research, even if we are not doing in a lab context, even if I have not done any study in my controlled way. So anyway, I think it is already 50. I have to stop the discussion here. You can please pose your questions when I am taking this class.